Okay, we'll start growth, development, and genetics. So first we're going to do a little bit on growth and development and some terms that we use. So the first one is obviously development begins inside the uterus. So for that we need to get pregnant. So pregnancy is from the time of fertilization till the child is born. So that is pregnancy. And the developing offspring is called the conceptus, and actually we'll see that this is further divided really into two stages. The offspring, we call it an embryo when it is till about 8 weeks. So this is actually further divided later, we'll see, into an embryonic period and a fetal period. The embryonic period is till 8 weeks and the fetal period is from 9 weeks till until birth. The period of gestation is the period of development within this pregnancy. And then how do you calculate when the child is going to be born? So that is what is known as the expected date of delivery, so EDD. And this expected date of delivery is different for different species. So for us, for the human beings, we calculate it as either 280 days after the last menstrual period. So the first day of a woman's cycle is counted, we count from there. So that's the last menstrual period. So the first day that she begins her menstrual cycle, that's counted as day one. So if you count 280 days after that, that's when we can say this is the expected date of delivery. We can never absolutely guarantee it because a child can be born, 10 days before, 10 days after, right? And 280 days is going to be a very difficult number to calculate. So this is easily done by 9 months plus 7 days. To her if you add 9 months and 7 days to her last menstrual period, which is called LMP, last menstrual period, you can very easily tell a woman what her expected date of delivery is. Or another way of calculating it is 266 days after fertilization. But we don't know exactly when fertilization occurred. So it's very difficult really to calculate that. So, we get a good estimate using this 280 days after the last menstrual period. Then development is of two types. There is before birth, which is during this gestation period when a woman is pregnant. But we have a lot of development which occurs after birth. If you remember when we were doing the respiratory system, I told you a lot of the alveoli develop up to about 8 years after a child is born. So you can see that development, some of it continues to occur after birth as well, which is known as postnatal. This is showing you like an early development from the time of fertilization. So you can see after fertilization, which should occur, after the oocyte has been ovulated, then you can see what's the size of a 1 week conceptus, a 3 week embryo, 5 weeks. Look at the embryo at 5 weeks, looks pretty, has kind of got a good, you can get a good idea that it's got a human shape here. An 8 week embryo, look at this 8 week embryo, perfectly formed. So after this, we start calling it a fetus. So up to by 8 weeks, almost all of the organ systems have formed. They're just not functional at that point. But you can see the limbs have formed perfectly, everything, eyes, head, and so on. And after this is when the growth of all these systems begins to occur in the fetal period. Now here is an example. Suppose you had to calculate somebody's last date or expected date of delivery, and I tell you that her last menstrual period was January 1st. And as I said, you always say it from the first day of the cycle starts. So what would her expected date of delivery be? Okay, it's October 8th. Now think of it, after January 1st, so you add 9 months. So, February, March, April, May, June, July, August, September, October 1st would be 9 months. And add 7 days to that. So that's October 8th. So that's why October 8th is the correct answer. Okay, so 9 months and 7 days. That's what you're going to add. Let's look at the period of fertilization. You can read some of these things. One, obviously, fertilization occurs when the sperm penetrates the oocyte, and then, at that point, the chromosomes of the male and the female chromosomes will meet together, and you'll get the zygote formation, which is the fertilized oocyte. As I told you when we were doing the reproductive system, the ampullary portion of the fallopian tube is the one where most often fertilization occurs. Oocytes tend to live for only 24 hours, whereas sperms live a little bit longer, 24 to 48 hours. So the timing, if a couple wants to get pregnant, the timing of the intercourse becomes very important, at least, 2 days before or after ovulation, so that, if, say, the ovum is liberated, so if it occurs immediately after ovulation, it's there, the sperm lives longer, so it can kind of, fertilize it, or before, so the sperms are present over there. The male produces, as we saw, 50 to 100 million sperms per ml. But of those millions, only about 50 or 100 actually make it to the ampullary region. Many of them kind of die in the way. Either some of them, immediately sort of get flushed out of the vagina, or they get, they're not able to survive the acidic medium, or they're not able to find their way, or they may be phagocytosed, and so then by the time the sperms actually reach the fallopian tube, only 50 or probably 100 of those million, only so many kind of reach, which is why the numbers are so many when they are produced. Also, when the sperms achieve maximum motility in the female reproductive tract. Remember that in the epididymis was where they were maturing. That was where they were, getting their motility. But absolute motility is achieved in the female reproductive tract through a process which is known as conditioning. And what happens in conditioning is certain changes occur in the sperms over a period of a few hours. The sperm has kind of got it's got a head part like that. And this is a little cap, and let's say that's the head. And then here is the neck, and this is the tail. So this part of the head is known as the acrosome. So here, the protective acrosomal membranes, they kind of get broken by enzymes produced in the female genital tract. This is also one of the reasons why whenever people go for assisted reproduction, imagine a couple is not able to get pregnant and they use resources outside the body. In that case, some sort of a fluid has to be added to the petri dish where the sperms are, which help in this conditioning. Because, as we see that this occurs in the female tract, but there the fertilization is occurring outside the body, so you have to do something to condition those sperms. So one is that this acrosomal region, this is kind of made fragile by the secretions of the female tract. And the idea is that the oocyte, when it's liberated, we'll see in the next few slides, it has a collection of cells around it. And these cells look like a crown. So these are known as corona radiata cells, because the oocyte has a kind of a membrane around it called zona pellucida. So the zona pellucida is, if this is the oocyte, there's kind of a membrane around it called zona pellucida. And then you have these cells which surround it, and these cells are known as corona radiata. So you can see that in order to get into the sperm, into the ovum, the sperm has actually got to pass through this thick layer of cells. It's got to pass through the zona pellucida and then get in. So you can see that that's why you need to it has to find a pathway through. That zona pellucida is like a protective covering, because what happens is it keeps the oocyte contained in a structure. And you'll see, which is why as the oocyte keeps traveling through the fallopian tube, the zona pellucida remains for some time. Because once the oocyte is fertilized and it begins dividing, it forms a lot of cells. These cells are highly erosive. So if that zona pellucida didn't contain the oocyte, what would happen is these erosive cells would start sort of gnawing in the fallopian tube itself, so that the zygote would sort of embed in the fallopian tube, which you don't want it to happen, right? So this zona pellucida kind of is a protective covering, and at about 5th day to 6th day after fertilization is when it disappears, by which time this zygote has actually reached the uterus, and then it can implant in the uterus. So these sperms, when they are conditioned, they also have to be able to pass through this layer of cells called the corona radiata, and they also have to pass through that zona pellucida to get inside and actually meet with the chromosomes of the oocyte. Once it touches the zona pellucida, the acrosome releases some enzymes so that it is able to kind of make little holes inside the zona pellucida, and it can enter into the oocyte, and then the oocyte becomes impenetrable to other sperms. So only one sperm is allowed to enter, just immediately becomes impenetrable to other sperms. The moment the oocyte is fertilized, only then does this secondary oocyte complete its second meiotic division. Remember, we were talking about that when we did oogenesis. If no fertilization occurs, let's say a woman, the egg has been ovulated, no fertilization has occurred, the secondary oocyte will not complete its second meiotic division, it will just pass out of the body like that, okay? The other important thing to remember is sometimes women know that, okay, this is about the time that my ovulation is occurring, and let's say that, they didn't intend to get pregnant, but they were sexually active around that time, but they're worried, am I pregnant or not? They don't want to wait for 2 weeks, to find out through a urine test whether HCG is being produced. So actually, immediately after fertilization, within 24 to 48 hours, a protein is present in the blood, which is known as early pregnancy factor. So this, if a woman knows that probably she could have gotten pregnant, she can actually get a blood test. And this is the basis of very early pregnancy tests. So, within 24 to 48 hours after she has had intercourse and she thinks she may have gotten pregnant because she was probably ovulating at that time. Remember, ovulation occurs 14 days before the next cycle. So if she has very, very regular cycles and she feels, this is the time I might have ovulated, she can actually check by doing this blood test. After fertilization, obviously, the diploid number is restored because the gametes were haploid. And sex determination is done by the father's sperm, because remember, the woman, when she produces an oocyte, it can only be X, because a woman, remember, was 44XX, right? So all oocytes have just the X chromosome, whereas the father was 44XY. So he could have a Y sperm or he could have an X sperm. So if the X and X meet, what will happen? It will be a baby girl. If the Y and X meet, then it will be a boy. So the determination of sex is determined at fertilization, and the father's sperm is the one. So the father is the one who actually determines the sex of the child. Now, usually only one secondary oocyte is liberated at ovulation, and I believe I mentioned earlier that it occurs alternately. So 1 month, say, the left ovary will ovulate, and 1 month the right ovary will ovulate. So therefore, usually it's only one oocyte which is fertilized. But people do have twins, they have triplets, sometimes they have quadruplets and so on. So how does this occur? So we have two types of twins, or use this for triplets, multiple births. One is known as fraternal and the second one is known as identical. Fraternal occurs when more than one oocyte is fertilized. So obviously more than one oocyte is liberated at ovulation. So therefore, if it gets fertilized, then if two oocytes are liberated at ovulation, then if they both get fertilized, so then you'll have twins. If three were, liberated, you would get triplets. This occurs a little bit more rarely. The more the number of multiple births, the more rare the occasion is. So, usually it might happen in women who are given, drugs to kind of, their reproductive cycles are not working well. So you give them these drugs to kind of enhance ovulation. So at that time, because that fine tuning doesn't occur, so there may be many ova which are liberated, and then that could get fertilized. Identical twins are twins which are twins or multiple births are produced where one zygote is formed. this zygote begins to divide through a process called cleavage. Cleavage occurs when the cell size doesn't increase, but the number of cells increases. So you have two. So if each one forms a individual embryo, you'll have identical twins. You can understand the sex will be the same, all the genetic material will be the same, right? Because it's from one zygote. Say this goes to a process where it's forming 3 cells. If each one divides and becomes an individual embryo, you can understand that you'll have triplets who will be identical, okay? So here is a slide showing you how sperm will penetrate. So you can see at this point. So this is the oocyte. Here's the zona pellucida, and here is the cell surrounding it here, which is known as corona radiata because it looks like a little crown. And here are many sperms which are coming. So here is the sperm which is coming close to the zona pellucida, releases those enzymes, and so that it can kind of, pass through. So here it passes through the zona pellucida and enters into the oocyte. And the moment it comes here, its nucleus is released, and then the zona pellucida becomes impenetrable to other sperms. So it doesn't allow the other sperms to enter inside up here. Okay, so that's how polyspermy is prevented, because you don't want more than one sperm entering, because what will happen? You'll have more chromosomes inside, right? Then the race will become absolutely different. It won't be a human being. And here are some of the events which occur after fertilization. So here we see the sperm nucleus has passed through into the oocyte. The moment it sort of passes through the zona pellucida, the oocyte's been fertilized. The secondary oocyte completes its second meiotic division. Then the nuclei begin to swell, and they are known as pronuclei. They kind of come together, join each other, and fertilization is completed at this point. The DNA and the chromosome number is established, and now this cell, this zygote, will wait for some time, for about 30 hours, and then it will begin to divide so that it can begin to start the developmental process. So think about this question: why does only one sperm fertilize a secondary oocyte? Okay. The zygote doesn't begin to divide as soon as fertilization occurs. It waits. There's a resting period, waits for 30 hours. Remember, I just said. So it is because the oocyte is permeable to only one sperm. Sometimes imagine if two were, it was like a photo finish. If two sperms sort of did everything at the same time and they got in, what would happen? You would have two sperms enter inside, right? So then you would have the number of chromosomes will be more, so that zygote would not be viable. So it would just be flushed out. So it never even goes beyond that. So you may have times like this when you might have, because if say many were to, theoretically, if some, 3 or more were to kind of go at the same time and they all enter, the zygote is not viable at all. It just gets flushed out, which is why we never have cases where you have more chromosomes. So here, as I was saying, the embryonic period is up till the first 8 weeks of development. This is an extremely important period because this is the time when organ systems are developing. And this is the time when anything which can affect fetal embryonic development, any drug or any environmental pollutant even can affect development. This is the time when they mainly cause damage. Such substances are known as teratogens. So teratogens are either drugs or radiation, something like radiation, which can affect the developmental period. And they actually came across this in the most odd manner. There used to be a drug called thalidomide, which is still there. And thalidomide, and I believe it was in Germany, I may be wrong about that, but I think it was in Germany. At one point, they found that a lot of children were born with what was known as phocomelia. You don't need to write this. This is just out of interest. So these children were born, and what they had was, instead of having limbs, they really had flippers. You know, the limbs were not properly developed like this. So they just had kind of little small appendages. So they really looked like flippers. So they went back and it was kind of during a period, they found a lot of children being born like this. And so they went back and they took the history and they found that this drug, thalidomide, was given to women for morning sickness. So, you know, morning sickness usually occurs during the early months of pregnancy, the first 8 weeks or first 3 months. And so all of these women who had these babies had taken thalidomide. And then they went back and they found that thalidomide was a very, very potent teratogen. So it affected the development and it acted, in this case, in limb development. And the children had other problems too, you know. So that's when they found that that's why now a pregnant woman is told, other than maybe taking folic acid, which is important for preventing neural tube defects, a pregnant woman is told to avoid any sort of drugs during the first trimester of pregnancy, which is the first 3 months of pregnancy, because this is a time when the organ systems are developing and you do not want anything to act in an adverse manner and cause problems. Okay. The fetal period is from 9 weeks till birth. So this is the time when the organ systems begin to mature, they increase in size, and their functioning occurs at this point. Though you will see that many of the organ systems begin to function even during the embryonic period. Some of the early sequence of events. So as I said, the zygote kind of undergoes a resting period, 30 hours to 36 hours. So after about 30 to 36 hours, the zygote undergoes a process of division which is known as cleavage. Cleavage is different from other division because here the cell size doesn't increase. So if you have a cell like this, it keeps dividing, it just gets smaller and smaller. So you get many cells, but each cell becomes smaller and smaller. So that is what is cleavage. By about 8 days, you get a ball of cells which is known as the morula. At this time, the embryo, this fertilized zygote, is actually moving along the fallopian tube. You know, the cilia of the fallopian tube beat and they set up currents, so it helps to move it along towards the uterus. So the zona pellucida also slowly starts breaking down. So it doesn't break all together. It slowly starts breaking down. So because of the columnar epithelium of the fallopian tube, which produces secretions because there's fluid in the fallopian tube, and that fluid current is what the cilia kind of set up and allow the zygote to kind of be propelled towards the uterus. Some of this fluid kind of enters the morula and it forms a little cyst inside. So imagine you have a ball of cells. So this is a ball of cells like this, and now in this ball of cells, the cells get kind of pushed over to one side, and this area is a fluid-filled cavity. So because of the presence of this fluid-filled cavity, you can see that the whole sort of ball has two parts. You have one part which is peripheral up here. That part is known as the trophoblast. It's on the outside. And then you have a mass of cells which are kind of concentrated towards one end. That mass is known as the inner cell mass. Both these regions of this whole structure, which is called the blastocyst, because there is this cyst-like cavity inside, it's called the blastocele. So that's why this is called the blastocyst. And actually all of these cells are known as blastomeres. So all of these little cells which are formed are known as blastomeres. So this blastocyst has two parts to it. One part called the inner cell mass, which is kind of concentrated at one end, and then one part which kind of goes all the way around like this in the periphery, and that's called the trophoblast. Each of these two parts has separate functions. The inner cell mass is the part which gives rise to all of the embryonic tissue. So the entire human being is formed from the inner cell mass. This outer cell mass gives rise to everything that does not belong to the embryo. All of this is shed from the body. So it gives rise to what is called the placenta. The placenta is shed from the uterus, and what are called fetal membranes. These fetal membranes are something known as chorion and another one known as amnion. So chorion and amnion. So this blastocyst sort of forms around the 5th day. It gets slowly kind of rolls along, and by day 6 it usually reaches the uterus, and that's when it implants. So the blastocyst implants or embeds into the uterus by day 6 after fertilization. Day 6 is always after fertilization. So you can understand, remember I told you last time that the fallopian tube is so narrow, it's just 2 hairs breadth. Anything which will delay the passage of the blastocyst through, what will happen? It will make it stick in the fallopian tube, and that's when an ectopic pregnancy can occur. It can embed in the fallopian tube. So here you can see, so here is ovulation. So this is the oocyte. Now it comes into the ampulla. Meanwhile, sperms have traveled up from here. So fertilization occurs in the ampulla and the zygote is formed. So, about 36 hours after fertilization, it begins to divide. You can see 2 cells, 4 cell stage. It forms a ball of cell. It's still slowly traveling through the uterus. So this is the morula, which is a ball of cells. And this, you can see the cell size. This is the initial size. Notice the cells just get smaller and smaller. The size of that structure doesn't increase. Each of these is known as a blastomere. As it's traveling through, fluid kind of enters into it. So now it gives rise to an area of cells at one pole, and then another area which kind of completely surrounds this cavity and this. So this outer area is called the trophoblast, and this inner area is called the inner cell mass. So this inner cell mass is what gives rise to the embryo proper, and this outer cell mass, or what used to be called the outer cell mass and is now called trophoblast, that's what gives rise to the placenta. And here you can understand, when it shows you 4 cells, but look here, it's 2 cells. So at this point, if these 2 decide each one to form a separate embryo, you can understand that this would be identical twins, because all the genetic material would be the same. So let's see how it goes in, and you can see how it's going into the uterus. It usually, implantation occurs in either the fundus or the lateral walls along the posterior side. That's where implantation occurs. Sometimes if it is hurried through and it comes lower down, it can implant here in the cervix also. That also is an example of an ectopic pregnancy. Okay. So let's look at implantation.
At the start of the menstrual cycle, a cohort of follicles starts to grow, but generally only one continues to develop. This follicle is referred to as the dominant follicle. The follicle contains an oocyte. This follicle increases inside by 1 per day. The follicle produces estrogen, causes the lining of the uterus, the endometrium, to thicken. Around day 12 to 14 of the cycle, the follicle has grown to the size of about 21. Rupture of the which is called ovulation, and your ovule is released. The fallopian tube's fimbriae move towards the ovary and catch the oocyte that stays near the ampullary zone of the tube, waiting to be fertilized. As the semen starts, its ascent tube to arrive at the oocyte. One of these will succeed in penetrating into the interior of the oocyte. One day following fertilization, a cell with 2 nuclei is observed, at which point in time it is called an embryo, and it begins to divide itself. 2 days after fertilization, the embryo is made up of 2 to 4 cells and moves along the tube in the direction of the uterus. At the end of 4 days, the embryo continues to divide itself, at which point it is called the morula, and the interior of the embryo has an area filled with liquid. This is called blastocyst. The blastocyst migrates to the uterus and sheds the membrane, process of hatching, and the embryo begins to implant in the endometrium of the uterus.
So let's see what happens after this. So here now we are going to look at, after implantation, we'll first look at how the placenta is formed. So first and most important, it's formed from the trophoblast. That's what forms the primitive placenta. This trophoblast has a very erosive quality. It can kind of gnaw and send out feelers. So it goes into the uterine endometrium and then inside the uterine wall. And what it does is it forms these little processes, like they look like villi. So these are known as chorionic villi. And later you will find that fetal blood vessels actually grow into this villi. And here, these blood vessels are surrounded. So you have fetal blood vessels which are present. So imagine fetal blood vessels, arteries and veins. And here is part of this trophoblast. So here's the trophoblast around it. So this is all cellular. And then the maternal blood surrounds it. And the maternal blood here surrounds it. So you can understand that's what is meant by bathed by maternal blood. This maternal blood is separated from the fetal blood by, can you see this area here? So there is no mixing of maternal and fetal blood. They don't mix together. They're always separated by this sort of membrane which is known as the placental membrane. This placental membrane, like anywhere else, remember we did the respiratory membrane, then when we did the urinary system, we did the filtration membrane. Same way, this placental membrane is also very thin. But this is what helps exchange of gases, because this maternal blood, which is kind of bathing it on the outside, from this the fetal blood vessels will take oxygen, and the carbon dioxide from the fetal blood vessels will go into these maternal sinuses and then be taken out, pass through the mother. So they are separated by this placental barrier, which is really, really important. The uterus, that you remember in the secretory phase, the cells of the uterus were kind of storing glycogen. That was important because those cells are the ones which are going to provide nutrition to the growing embedded zygote and embryo inside. So that's where it gets its nutrition. The other thing is this trophoblast secretes a very important hormone which is known as human chorionic gonadotropin, or HCG for short. This is excreted in the urine of the pregnant mother, and about a week or maybe 2 weeks after fertilization, which is about the time that she first misses her period. Let's say she doesn't know she's pregnant. 2 weeks after fertilization is 2 weeks after ovulation has occurred, right? So that's the time when it is time for her next period to occur. So, when she has her first missed period, around that time is when HCG amounts are enough produced so that you can do an early pregnancy test, and that's what the pregnancy kits, that's what you use for, the ones, which test a mother's urine. The other important function of this HCG is that it keeps the corpus luteum in the ovary, it keeps it functioning, because the corpus luteum is very important. What hormone is produced by the corpus luteum? Progesterone. Remember, progesterone was very important to maintain pregnancy because it keeps the uterine lining soft and moist, causes it to retain, and glycogen causes the glands to secrete. That's why that phase was secretory, so that all this developing embryo can be nourished by this moist environment. Then after about 2 to 3 months, the placenta itself takes over, so it is able to produce progesterone and estrogen. And they say actually that within the very early weeks of pregnancy, if you were to kind of remove the corpus luteum, the pregnancy would sort of end. But after about 3 months, if you take out the corpus luteum, it doesn't matter, because now the placenta itself begins to produce progesterone and estrogen, so it can help. It kind of acts as the lungs and the kidneys for the fetus, so it removes waste substances and carbon dioxide, and it provides oxygen and nutrients to the fetus. Then again, as I said, the cells of the trophoblast, not only do they form the placenta, remember they form the fetal membranes also, so they form this called the chorion and amnion. And this amnion, we'll see in the slide, it lines a cavity which is known as the amniotic cavity. This is what you all probably know of the bag of waters, which when a pregnant woman goes into labor, we say the bag bursts. So this bag of waters is nothing but this amniotic sac. So it keeps increasing in size. As it keeps increasing in size, it kind of surrounds the fetus totally. So it forms a nice environment, like a nice water bath for the fetus, so it acts as a shock absorber. It also is an area which allows fetus to move, so fetal movements occur. Whenever amniotic fluid is too little, what will happen? Fetal movements don't occur, so the fetus could be born with where areas where the limbs are kind of stuck to each other, like, so they are conjoined. So you need enough amniotic fluid. 600 ml is about the amount which is normal to allow fetal movement. So if the fetus is able to move properly, then it can grow properly. Sometimes when the amount of amniotic fluid is less, fetal movements don't occur, and a lot of congenital problems will occur at that time. Things will not grow properly. You'll find that the limbs may be attached to each other, and you get other problems. This amniotic fluid is produced by the cells which line it. Now, again, like anything in the body, you can't keep producing amniotic fluid and not removing it, right? So what helps to remove it is actually the fetus does. So the fetus, since the amniotic fluid, into the amniotic fluid, the fetus actually passes urine. So that adds to the amniotic fluid. But the fetus helps to remove the amniotic fluid by swallowing it. So it actually swallows and inhales it. So it passes into the body. And that's how it helps to kind of keep the amount of amniotic fluid more or less constant. Sometimes when the fetus has some nervous system problems, like say the brain is not developing properly, the swallowing center is present in the brain. So that if the brain doesn't develop properly, the fetus will not be able to swallow because the swallowing center is not proper. So what will happen? The amniotic fluid will keep on being produced, but it's not being sort of taken in by the fetus. So the amount of amniotic fluid increases. So sometimes that condition is known as hydramnios. And sometimes when women, that's one way actually an OBGYN can diagnose that. When she sees that a woman has too much amniotic fluid, they immediately start thinking, is there a problem with nervous system development? And is that why the amniotic fluid is more? I mean, there could be other reasons why, but this is one of the reasons why it occurs. This amniotic sac, like I said, is the bag which actually during labor, it is the one which pushes against the cervix. It helps in labor, and this is the one which ruptures and, actually begins the process of labor. So this was the trophoblast. The inner cell mass, that's the one which gives rise to all the embryonic tissue. So in the beginning, you noticed it was just a group of cells which were present at one end. But by the end of the second week, these inner cell mass, they kind of line up in such a way that they form two layers. So you have two layers of cells. You'll have one layer on the top and one layer at the bottom. So that's what is known as a bilaminar disc. So two layers. And later we'll see that these two layers will actually become three layers, and that's what gives rise to the three main germ layers which give rise to all the organs of your body. So here let's look at this formation of the placenta. So here is the blastocyst. So you can see this blastocyst up here. So this was the area where the inner cell mass was. This one is the trophoblast, and the trophoblast also gets kind of divided into two parts called syncytio and cytotrophoblast. Notice how this trophoblast is growing. So we'll first look at it. Here are the maternal blood vessels. See how the trophoblast is increasing, and look at here, the embryo has just implanted little bit. Now it's pushed more inside. Can you see now it's gone further inside? And what it does is it nears these blood vessels, and here you can see this. This is where the primitive placenta is established. At the same time, look at this inner cell mass. Notice how it forms this bilayered embryonic disc. So again, this is bilayered. And then a third layer grows in by the end of about close to the third week. You have the third layer formed. So you have three germ layers formed. These three germ layers are ectoderm, mesoderm, and endoderm. And these are the ones which are going to give rise to all your body tissues. So these three germ layers come from the inner cell mass. So it's the inner cell mass which gives rise to the three germ layers. First there were two. So here there were two layers. Here also two. A third layer insinuates itself in between, and you get three layers. And that's what will give rise to the inner cell mass. To all of the embryonic tissue. In the beginning, what happens is, can you see that the placenta is all over the place? So it completely kind of surrounds the embryo. Here's the developing embryo. It completely surrounds it. After some time, this part of the placenta will disappear, so that's why we have the placenta only developing at one end, and it is connected to the growing embryo through the future umbilical cord. You all know that the fetus is connected to the placenta by the umbilical cord. So here is the area where the umbilical cord will develop. So here you can see, so this is showing you, it'll grow. So here notice how the placenta is only at one point connected to the embryo through this umbilical cord. And here you can see this is the part where, this is the amniotic sac, which is completely surrounding the fetus and embryo. So notice how it's kind of lying in a water bath, which can act as a shock absorber. So if the fetus urinates, the urine will pass into the amniotic sac, and through this umbilical cord you can see that the nutrients and oxygen will go to the fetus, and carbon dioxide and waste will pass out. Because of this fluid, it keeps the fetus and embryo buoyant. It also allows movement so that the uterine walls don't close in on the developing embryo. And here you can see at this point, this is showing you, these are the maternal blood vessels. So both arteries and veins open into a sort of dilated structure, which is called maternal sinus. Here are the fetal blood vessels. And look at this. This is the tissue, that trophoblast, which surrounds these fetal blood vessels. So these are known as chorionic villi, and this is the placental barrier. So oxygen from the mother passes through these cells, through the endothelium and into the fetal blood vessels. Carbon dioxide from the fetal blood vessels passes out and will be taken by the veins and go back. The mother will take care of it. Also, this is another thing you've all must have heard of a procedure known as amniocentesis. If you haven't, amniocentesis is a procedure where you can remove amniotic fluid. Now, why would you want to remove amniotic fluid? Anybody tell me? Why would you want to remove amniotic fluid? Pardon? You can do genetic testing, yes. See, notice how the fetal skin is kind of bathed by this amniotic fluid. So like we shed our skin cells, the same way the fetal skin cells are all shed, and they will come and pass out here into the amniotic fluid. So now when you do an amniocentesis and you take amniotic fluid, you can understand you'll get some fetal skin cells in that. Then you do testing of those fetal skin cells. You can see if there are any chromosomal abnormalities. So you can do genetic testing. I mean, you can also tell obviously the sex of the child, but that's not why anybody would want to do it, because it is with its own problems when you do that. You can kind of start labor and you can cause bleeding. So you only want to do it when there is some problem. So you can do genetic testing like this. You have another testing which you can do where you can actually remove cells from this placenta called chorionic villus sampling also, because that's part of the same thing, part of the embryo. So you can test for genetic, if there are any genetic problems, chromosomal abnormalities, and say the parents don't want to go ahead with pregnancy. You always will want to do amniocentesis earlier in the pregnancy when it can be aborted. Okay? So let's look a little bit at the formation of the placenta. So there's no audio here. So here you can see the blastocyst. It's come into the uterus. It begins to push through the endometrium, so it's empty. So these are the endometrial cells. So it's passing through. And this, which, like I said, it's very erosive, so it kind of, eats into the endometrium. Meanwhile, notice the inner cell mass is slowly also forming two layers. These are uterine glands and blood vessels. So the two layers, here's the amniotic cavity which is forming. And opens up glands, so the secretions of the glands can directly nourish the growing embryo. and now it's just getting larger. Maternal blood vessels have been eroded, so they are pouring blood into spaces which are known as maternal sinuses or lacunae. Meanwhile, here the embryo is also growing. You can ignore all these other things, which form. So here there were two layers initially. As the embryo begins to grow, a third layer will be added. Those are shown. They do.
Amniocentesis is a procedure that lets doctors detect or rule out problems with your baby's health long before birth. In the uterus, your baby is surrounded by amniotic fluid that provides protection and nourishment. Floating in the fluid are cells from your baby, providing doctors with genetic material testing. In amniocentesis, your doctor will take a sample of this fluid to test for specific genetic disorders, birth defects. Amniocentesis is done in the second trimester of pregnancy at 15 to 18 weeks. It is generally offered to women with certain age 35 or old, increases the risk of disorders, Down syndrome, a previous child or pregnancy with a birth defect, blood test or ultrasound suggesting a birth defect, such as Down syndrome or neural tube defects, or a family history of genetic disorders, such as cystic fibrosis. Amniocentesis may also be done in the third trimester for these reasons: to determine if the baby's lungs are mature enough should early delivery become necessary, to diagnose a uterine infection, or to check for anemia in a baby with Rh incompatibility. To begin your procedure, your doctor would use ultrasound to create an image of the fetus and nearby structures on a computer screen. Looking at this image, the doctor can choose a safe place to insert the needle. Your doctor may give you an injection of local anesthesia to numb the surface of your abdomen where the amniocentesis needle is to be inserted. Carefully watching the ultrasound monitor to avoid contact with the placenta or the baby. Your doctor will introduce the thin needle through your abdominal wall into your uterus and into the amniotic sac. He or she will then withdraw a small sample of amniotic fluid, remove the needle, and cover the site with a bandage. Your body will make additional fluid to replace what was removed. Your doctor will continue to monitor the ultrasound to check that the fetal heartbeat is normal and the tolerable. Feel cramping or pressure lower abdomen. For about 24 hours after your procedure, you will be advised to avoid physical stress, such as vigorous exercise, lifting, prolonged standing, and sexual activity.
Okay, let's look at, let's review some of these some of what we've done. So what is the structure that is responsible for maintaining corpus luteum? What is that structure formed by? I don't know, did it show? Well, for some reason it didn't. So anyway, the correct answer is the trophoblast, because trophoblast forms the placenta, which is responsible for maintaining corpus luteum. Okay, let's look at this one. HCG, yes, very good. The early pregnancy factor is not formed by the trophoblast. That was after fertilization a protein which is formed, okay? And this HCG is the one which you test in the urine, the off the counter pregnancy tests. Okay, a couple say do not use contraception, and what would be a way to establish if she is indeed pregnant? Early, very early pregnancy. EPF in the urine. Remember, this is HCG in the EPF, in the blood. HCG in the urine is 2 weeks after fertilization. So, she's already 2 weeks pregnant. So that's not really early. This is the one which is within 24 to 48 hours she can get a blood test to tell. And is there a mixing of fetal and maternal blood during pregnancy? No. Okay, so here now after this, the germ layers, from a flat, initially it's a flat slipper shaped, the embryo is absolutely flat. And we saw 2 layers are there, then the 3rd layer gets formed, and you get these 3 layers, and you can read, the structures that they form. Nervous system begins to develop. The time when the nervous system develops, that time is called neurulation. The cardiovascular system develops rather early, by about, 3rd week, 21 to 22 days. The heart, you can see the heartbeat and an ultrasound at that time. All organ systems form during this embryonic period, but the function is minimal. Also, in the beginning, the embryo is absolutely flat. Then it begins to fold. So, imagine you have a flat plate like this. So if you're looking from the top, you have a flat plate like this. This folds in this direction. And it also folds like this. Because, we all know we have a body cavity inside us, right? So we are not flat, isn't it? And that folding occurs. And that folding leads to the formation of the gut tube. Also at this time is when the limb buds become visible. So when you start looking at the embryo, you have tiny little flippers which form. They kind of extend and can finally become the limb. At this time, that's why you need enough amniotic fluid, because the limb movements begin to occur. The mother cannot feel the limb movements around this time. That happens much later. And by about 8 weeks, you can see the kidneys have begun to function. So urine begins to form, and that is, passed out in into the amniotic cavity. And this is the time when teratogens, or very harmful substances, actually cause problems within the embryo. From 9th week onwards, which is what is the fetal period, the organ systems begin to grow. The woman's sort of abdomen gets much larger. The entire pregnancy is divided into 3, what we call 3 periods, or what are known as 3 trimesters. So we say it's about 9 months, right? So we have the first trimester, the second trimester, and the third. The first is from 1 to 3, this is 4 to 6, and this is 7 to 9. Okay? So this is months. Trimesters are in months. So, obviously by about 9th week is 2nd month onwards. By about 12 weeks, actually, the uterus kind of begins to reach the pubic symphysis, starts, a mother can then she begins, in the 2nd trimester onwards, the woman looks pregnant. You can see a bump. Fetal movements, these are movements which the mother can actually feel. These movements are known as quickening. They're usually felt by about 17 to 20 weeks of pregnancy. The first time a woman is pregnant, she might feel it later, like around 20 weeks. But after that, 2nd pregnancy onwards, she knows what to expect. So usually she starts feeling them about 17 to, 17 or 18 weeks. Surfactant production, by about 24 weeks, it starts being produced at that time. It keeps increasing, good amounts are produced well after 28 weeks, which is why you want pregnancy to continue and a child to be born full term. So here you can see, so this is, it's showing you the bilayered amniotic disc. So then this becomes tri-layered. So this is a flat disc at this time. It then folds. So can you see it's kind of folding from this end? It also folds on the side. So that's how you get the formation of a gut tube. So let's look at this. This is kind of difficult to appreciate unless you see it in 3D.
In this video then we'll look at the beginning of development, looking into the ovary at an unfertilized egg. It then gets fertilized by sperm travel down, coming along. So will hit the egg and try to penetrate it, but one will win, as it were, go into the nucleus, and then there's a reprogramming process that male their genes aside to be turned on and off for early development. Here you see early cleavage stages occurring, and this is one of the early growths. The embryo moves down the fallopian tube. It's going to form an important stage called the blastocyst in a few seconds. Of course, in real life that takes days, about 5 days. At this stage then, I'd like to draw your attention to the inside of the blastocyst, where there are cells called the inner cell mass, which I'll be abbreviating as ICM. Those are the cells that make our animal. And the outer cells give rise to the placenta and other supporting tissues. At this stage, the embryo implants into the wall of the uterus. This is when a pregnancy is really initiated. And now those inner cell mass form its, and then as they continue to change their physical position, their kind of graphical relationship to one another. And you'll see that represented here as this disc gets transformed into an embryo. Those lines represent sites where cells are migrating in and out. And here's an important stage when the 3 beginning layers of the embryo, the so-called germ layers, are formed. And I'll come back to that in a few minutes.
Yeah, I just want to see flat and slipper shaped. Can you notice? It's absolutely flat discs. And now notice how it will fold.
As development proceeds, there's more growth and movement of cells. It'll begin to form a neural tube. Here it turns, and appendages start to bud out. You see the head forming and the eye, and then eventually we get a embryo, and some months later will be born as a young baby.
So you saw how it kind of folded, like being flat. You get all of these structures are formed. So this is just showing you from embryo to fetus, and notice how the gut tube is formed inside. From this gut tube, actually the respiratory system also gets attached. We did the digestive tract, and the urinary tract develops separately over here, the genitourinary part. The heart develops in this area, then, the eye buds and the ear buds, all of that develop. So let's answer again a few questions here. So what is the difference? The difference between fraternal and identical is that fraternal are always of the same sex. That's false. Yeah, it's identical, which are always of the same sex. Here are some changes which occur during pregnancy. One is the first one. A woman knows she's pregnant when she misses her period, right? So that's stoppage of menstruation. That is known as amenorrhea. Menorrhea is bleeding, amenorrhea, no bleeding. Pregnancy is usually about 38 to 40 weeks. And as I said earlier, we have 3 trimesters, meaning it divides into blocks of 3 months, first, second, and third. The uterus enlarges, and this is one organ in the body which is capable of, not only is there an increase in number, in the size of the muscle fibers, but increase in the number of muscle fibers. And that increase in number of muscle fibers is known as hyperplasia. Also, as the uterus sort of grows, the height changes. Like I told you, I mean, you don't have to remember these things, but by about 12 weeks of pregnancy or 3 months, the uterus has kind of reached the pubic symphysis. It was well below in the pelvic cavity. It reaches the pubic symphysis by about 24 to 28 weeks, somewhere close to the umbilicus, and it keeps kind of increasing in height. Towards the end of pregnancy, it actually drops down a little bit because the fetal head comes down. So the height and that's why the abdomen kind of increases in size. Also, because the uterus has to become sensitive to oxytocin, the oxytocin receptors increase throughout pregnancy due to the presence of estrogen. Some women may have certain changes, such as, the skin may get pigmented around especially seen around the cheekbones. So this is known as chloasma. This pigmentation does not go away. The mammary glands increase. There's increase in the glandular part of the mammary glands. The placenta also produces this hormone called relaxin, which makes the ligaments extremely sort of lax. And so pregnant women also tend to have that, the typical walk, which is there. And also the growing uterus puts pressure on the back. So they complain of back pain. Usually about 30 pounds is the amount if it's a single pregnancy, in the sense no twins or anything. Almost all body systems are affected by pregnancy, either just the hormonal changes or due to weight of the uterus. Like the uterus presses on veins. So sometimes women, if they're standing for too long, they'll find edema in the legs. A gastrointestinal system gets a little bit irritable, the urinary tract, because of pressure. So a lot of it is to do with the pressure. And then finally, labor is a process where the child is, expelled from inside the uterine cavity. That other thing is the word for it is parturition or childbirth. So let's look at some of the stages of parturition. This occurs during the last few weeks of pregnancy. There's an greater increase in the number of oxytocin receptors, and this makes the uterus a little bit irritable. And in fact, some women undergo what are known as false labor pains, or also called Braxton Hicks contractions. So that's kind of false. But towards the final stages of pregnancy, when the bag of membranes and the fetal head kind of come down into the cervix and put pressure, that kind of sets on the onset of labor. So it's in 3 stages. The first one is from the beginning of uterine contractions to complete dilation of the cervix, because you want the baby to come out. This is the longest period and very painful. This cervical dilation is extremely painful, and that's what is responsible for the pains that a woman sees at this time. The amniotic sac kind of is pushed towards the cervical opening, the external os, and the head of the fetus presses against it, and usually that's what causes rupture towards the end of this stage. Stage 2 is when the fetus is expelled. So this is from when the cervix is completely dilated, which is about 10 centimeters until the exit of the fetus. And the last stage, also extremely important, is when you want the entire placenta to be removed. You don't want any bit of the placenta to be left behind, because once the placenta is removed, then the uterus begins to contract and it tries to come back to its normal stage. It doesn't remain big. And that's what will prevent the further bleeding. Otherwise a woman will continue to bleed if uterine contractions do not occur after the expulsion of the placenta. So here you can see the phases of labor. So this is when, look at this amniotic, the sac coming towards the cervix, the fetal head. So this pushes against it. So you can see as it's pushing against it, the cervix is kind of dilating. So till complete dilatation, that's stage 1. The fetus then passes out. So that's stage 2. And then here you can see the placenta coming out. So that's stage 3. So let's stop here.